for inviting me for this exciting symposium. So what I would like to do is to discuss a couple of the critical knowledge gaps in the malaria transmission cycle, work that we have been doing in the last few years to target these knowledge gaps, and then work that we have been doing to use the biology underlying these gaps to develop molecular tools to then look at infection and infectiousness in Plasmodium falciparum. So some of the questions that we have been asking is, where are gametocytes sequestered, and related to that, which gametocyte stages are actually present in circulation? And then ultimately I'm going to get at how long are gametocyte infectious and what determines infectious, and things that we are currently interested in. So where do gametocytes sequester? That's something I'm going to go into much detail tomorrow morning in another session at Symposium 173. But the basic answer is the majority of them is sequestered in the human bone marrow. And this was a large collaborative study with Terry Taylor's group in Blantyre, Malawi, and a number of other people, where we looked at autopsy cases of children who died of cerebral malaria, and then did a comparative analysis by immunohistochemistry and qPCR. And what we found is that not only are they sequestered in the bone marrow, but they're actually sequestered in the extravascular space of the bone marrow. This is a very important finding, and the first description of extravascular sequestration of blood stage malaria parasites. An important outstanding question in that study was, does gametocyte formation actually happen in the bone marrow? Does it happen directly on site here, or does it happen somewhere else? And that's important, because if it's happening here, then the only stage that we find in circulation of the gametocyte cycle in falciparum would be the mature gametocyte. If it happens somewhere else, and it's this young gametocyte that eventually gets here and sequesters here, then we would also predict to find very young gametocytes in circulation. And the two together would be very interesting to look at the dynamics of gametocyte formation in different contexts, such as experimental infections, drug treatments, or longitudinal studies. So to get at that, we collaborated with the group of Curtis Hottenhauer at the Biostats department in Harvard, and again with Terry Taylor's group and Danny Milner, as well as some of my former graduate students were involved and other people. And so basically the idea was, if we analyze in vitro microarray data which are published, so in vitro time courses from asexual and sexual parasites, and compare them with peripheral blood samples from malaria patients, again microarray data that's already published, and then look at the differential expression of genes across the life cycle, could we then determine which stages are present in circulation and which stages are absent, and then basically validate that by qPCR. So I'm not going to go into the entire transcriptional network analysis that underlies that, but basically, as a proof of principle, well, so in the first place, we identified about 285 transcriptional clusters of a size of at least 5 genes that are covering the asexual and sexual cycle of Plasmodium falciparum. 35 of these clusters are putative gametocyte specific. Gametocyte specific means that they're enriched in immature or mature gametocyte genes, as we defined them previously in a study, or that they're enriched in very early gametocyte genes, as we defined them in this study de novo, basically using 3 different attributes. One, a recent study that we did to flow sort sexually committed chysons. The second one, to look at co-expression of genes with a recently identified transcriptional switch, AP2G, that is exclusively expressed in sexually committed chysons and gametocyte rings. And then thirdly, a study that was recently published by Till Voss's group to look at the gametocyte induction in the absence of a heterochromatin protein. So what you can see here on the left is one of these mature gametocyte clusters, which contains a number of known mature gametocyte genes, such as PFG377 or PFS25. And on the next axis here, you can see in each column one patient microarray data point, here from a study in Senegal and here from a study in Malawi. And what you can see is that in a subset of patients, all these genes are expressed. So in 3 patients from Senegal, you can find all of them expressed, and in 4 patients from Malawi. Microarray data are not very sensitive for the detection of rare stages or rare transcripts. So of course this is an underestimate, but the good thing is these 4 patients here are also all positive by smear for gametocytes. And basically that just validates the point that we can define stage-specific transcriptional gene clusters, and we can detect all the genes from such a cluster in the blood circulation if this stage is expected to be in the blood. So now, if we do that with the gametocyte ring or the very early gametocyte clusters, these are 3 out of the 5 clusters we identified. They contain, again, known very young gametocyte markers like GEX5, PFS16, PF140748 or 744. And when we use, again, this AP2G marker as a positive control, because it's expressed in sexually committed chysons and early gametocytes, then we can see again that in a subset of patients across these 2 cohorts, all the genes or most of the genes from these early gametocyte clusters are expressed. So this demonstrates that at least transcriptionally we can detect the profile of very young gametocytes in the peripheral blood of malaria patients. And that's also the first evidence for gametocyte ring profiles and their detection in the blood. Therefore, so far no markers exist for this very early time frame of gametocyte development. PFS16, for example, is only turned on after about 30 hours of development. So it's very interesting, of course, then to use such very early markers in a qPCR assay to validate this finding. And we used these 2 PF140744 and 748 in a cohort of malaria patients, again from Malawi, where blood was collected. These are severe malaria children admitted to the hospital in Blantyre. And again, we can find the transcriptional profile of these 2 early markers in the blood as much as a constitutive marker and PFS25. We cannot detect that the high prevalence AMA1 as a Schizont marker or this immature gametocyte marker, PFS4845. So basically, we can use this knowledge now to develop a qPCR assay that covers the circulating asexual and sexual parasite, and that could then get at some of the questions that we want to ask in terms of parasite infection, gametocyte infection, and infectiousness. So this is a collaboration, and people have mentioned it before, with multiple people, including Diane Worth and Sarah Volkman and Caroline Bucky at our school, Terry Taylor's group again, and then Tom Boosma and Chris Grakely. And a number of students from different labs are involved. Here, basically the idea was to now use this knowledge and develop a qPCR assay that can be validated on field samples and in vitro, and then perform a proof of principle study. So this has been published before, so I'm not going to go in much detail, but basically we selected a marker for each of these circulating stages, the parasite ring, the asexual parasite ring, the young gametocyte, the mature gametocyte, and then the sequestered late asexual stage, and then 1 marker is a constitutive marker. So the gametocyte markers are absent in gametocyte deficient lines. This one is the one that Ingrid just mentioned before. And when we do in vitro time courses, the smear positivity or the counts of different stages correspond to the relative qPCR profiles that we can get when we do PCR. And then we can also infer the stage composition using a model that we developed in parallel with that. So basically, we developed a highly sensitive qPCR assay. This, again, like the one that Ingrid presented, amplifies across exon-exon junctions, so there's no danger that we also amplify from gDNA. So what we then wanted to do most recently is to apply it to a study basically as a proof of principle. So this study has been published, again, that's by S.E. Fulai et al. In Lancet Infectious Disease in 2013. And here the question was which dose of primaquine is efficiently knocking down gametocytes in the context of artemisinin lumefantrine treatment. That was done in Uganda. And so here, treatment with artemisinin lumefantrine was done at days 0, 1, and 2, and then primaquine was a single dose here. What you can see in this study, only PFS25 Q18-ASPA was done, and then parasitemia was detected by smear. And what you can see is that that upon primaquine treatment, gametocytes are efficiently knocked down in all of the 3 treatments. And here at day 14, they're at a very low prevalence in the population in the cohort. So what we wanted to do was to use this qPCR assay to basically confirm these data, to look at asexual parasitemia, and then also to correlate this data with genotyping information using the molecular barcode assay that was developed in DOWNLO's lab. So what we see is, using the immature gametocyte marker and the mature gametocyte marker, that ASTMH alone is not really hitting them, but adding primaquine efficiently knocks down the gametocyte prevalence to basically the level that Tone and colleagues have already noted using QT-NASPA with PFS25. But what we also noted is using the asexual marker. So this is it's SBP1, one of the most highly expressed transcripts in asexual ring stage parasites. Both artemisinin lumefantrine alone and artemisinin lumefantrine and primaquine treatment does not completely knock down the parasites from the population. So about 30% in both arms, each arm here that we analyzed was a cohort of 25 people, about 30% of them still have circulating asexual rings in the blood 14 days after initiation of the treatment. So this is quite surprising and suggests that there's a subpopulation of persisting ring stage parasites in a subset of these people. And so we estimated the parasite burden based on the slight positive samples from day 0 and the corresponding CT values, and then basically inferred the parasitemia at the later stage time points. And you can see that in all the patients, the parasitemia was significantly knocked down by more than 90, probably 95%. But then again, in both of these arms, this low-level parasitemia persisted in a subset of the patients. We also did molecular barcode, as I mentioned, and of course, these are all multiply infected people. But what we could see is that the diversity of the barcodes did not change between this initial time point 0 and the subsequent time points, suggesting that it's not that we are knocking out a major genotype or parasite population and we are left with a few chronic genotypes, but it seems like we are knocking down the majority of the parasites from all the genotypes, and we are left with multiple genotypes even at this very low prevalence. And then we also looked at a couple of parasite versus patient parameters, and we can see what can actually be anticipated, but interestingly, even in a cohort that's in a relatively narrow age range, so the presence or the load of the asexual ring parasitemia negatively correlates with age, so the younger the patients, the more parasites they have at the beginning. And the older the patients, the higher proportion, the proportion of the gametocytes, while the total load of gametocytes does not increase. And then corresponding with that, the younger the patient, the more likely to have persisting ring parasites, and the older the patient, the more likely to have persistent gametocytes, at least in the AL-only arm. So this, at the moment, is a very interesting finding using the tools that we developed recently. And of course, we now want to see whether these ring stage parasites persist beyond day 14, and there's later samples that we can analyze now, whether they serve as a reservoir for gametocytes, and whether these gametocytes are infectious. Unfortunately, it seems like if there are gametocytes, they are below our detection limits, and there were no feeding assays done. But it gets at the last question, which we really now starting to become much more interested, which is actually, so what is an infectious gametocyte? So now we know what's in circulation very early on, and we know what's in circulation very late, but we don't know which of these circulating parasites are actually infectious. So this is a graph that Lauren Childs from Caroline Bucky's lab generated based on published neurosyphilis data, but basically the point is, upon sporozoite infection, we know that asexual parasites start to appear after about 10 days, and then the mature gametocytes start to appear a little bit more than after 20 days, because in the meantime they sequester, and then they become infectious to mosquitoes. But of course, we don't know which of these mature gametocytes and at which point, once they are mature, they are infectious. We don't exactly know how long they circulate. And of course, we also know that there's other factors like the sex ratios, the genetics of the parasite, human immune responses that determine the infectiousness of a sample. But this is just now as the last slide here. There's some interesting things that we can get at the question of infectiousness. So this is a heat map that plots the gametocyte clusters here on the x-axis and the microarray data per patient from this study in Malawi that we analyzed before. And this is basically the mean expression across the cluster. And you can see, again, as I mentioned before, a subset of patients have persistent expression of these gametocyte ring clusters, and another subset of patients shows persistent expression of these mature gametocyte clusters. What we can also see is that clusters that apparently are present or specific for mature gametocytes do not seem to be present in peripheral blood samples from patients, suggesting that what we think is a mature gametocyte in vitro may not actually be necessarily one that's in circulation in a patient. So we need better understanding of what represents a mature gametocyte in vitro, or how we can measure mature gametocytes in a patient. In vitro, a mature gametocyte is at least one that's deformable enough that, or that's deformable so that we can measure, which represents one that can pass the spleen. In vivo, an infectious sample is probably one that infects a mosquito, while a non-infectious one is one that doesn't infect a mosquito. And these two cohorts could potentially be compared, again, by transcriptional profiling to get at the question whether there's some infectiousness markers in samples which are gametocytemic and infect versus those that are gametocytemic and don't infect. So with that, just a little outlook. Of course, now there is some questions that we're very interested in. How long are gametocytes infectious and what determines infectiousness? And then something that Tone was also mentioning, are mature gametocytes clustered somewhere, and not only in the venous blood circulating, maybe in some subdermal microcapillaries, maybe not even all the time during the day. So again, if we can close these knowledge gaps, then that can inform us for the development of new tools that ultimately get us at the question of the human infectious reservoir. So I'd like to thank lots of people from my lab, other groups from the school, of course, Terry Taylor and her group, Tone and Chris, and then Pietro and Kim Williamson, and of course the funding through the Gates Foundation and some other sources. Thank you.
Thanks very much, Matt. So it's paper's open for questions now. James.
Matt, lovely data. I'm intrigued by these late asexual markers that are present, and I'm certainly going to be wanting looking at that in my system. I just wonder if you've got
the early ones.
Sorry?
You mean the early asexual markers?
Yeah, the early asexual markers in my late blood stage challenge system, where I see obviously persistent parasite genomes and PFS25. My question really relates to the quantity of, have you done any estimates of quantity based upon your asexual marker of parasites at those late stages? Because you'll have, I expect, 18S data to be able to estimate parasite number and transcript numbers of your asexuals and your gametocytes. So have you done any calculations to try and figure out what
like I mentioned, I mean, we try to estimate it at the moment just based on the known parasitemia from day zero, but we are currently doing dilution series for cellular dilutions to actually then get at better numbers, because of course, if you estimate based on microscopic parasitemia, at the low end, there's a lot of noise. At the moment, we can't really tell how much these represent, but we want to find it out. I mean, the QT-NASPA assay with PFS25, as you know, probably detects about 0.1 gametocytes per microliter. That can't be seen at this late time, at day 14, so there's no gametocytes left, as it seems like, but we still can see these ring marker transcripts. So probably the ring marker is present at a very low level, and we have to find out which level exactly it is.
Thanks.
Matthias, about this persistent ring stages 2 weeks after treatment. So sounds like it's the dormants induced by artemisinin, right?
I don't know. I can't tell at the moment. We know, I mean, at least it's clear that this marker is not expressed in mature gametocytes, so we are not detecting low-level mature gametocyte signal. It's a population of parasites, because the RNA certainly would degrade if it's just something that's still floating around, but we don't know what the nature of these parasites is at the moment.
Would be very interesting to test them in slow clearers.
Well, I don't have these samples, and I don't know whether somebody has these samples.
So, Matt, in your late what you think are infectious gametocytes, which means there's a cluster that you see which correlates with that. What kind of genes do you see them? Are these related to energetics or related to any particular biochemical pathway?
So those clusters that we can detect, where we can detect the genes in peripheral blood from patients, they are just lots of known mature gametocyte genes. I mean, it's nothing unexpected. So there's one cluster, for example, is very specific for male egg flagellation. Other clusters contain lots of gamete proteins. So it's not something specific that we see. I mean, it's basically representing what we already knew about mature gametocyte transcripts, but many more, because there's lots of unknown genes in the same clusters. But I think to find out which subset represents an infectious mature gametocyte, we would have to compare.
Right.
Okay, thanks, Matt. I don't know if there are any more general questions from the floor, but if there are, we'd welcome. Okay, well, I'll take that as a no. And I'd just like to thank all the speakers in this session, and thank you for your participation and your questions. Thank you very much.